Life is full of choices. We make choices all the time. You're choosing to watch this video. I'm choosing to record a video for you. Throughout the video, I'm going to choose to draw some things, and I'll do it without much delay. The ability to make choices quickly and efficiently is probably something that we take for granted. And it's not until you see something like Parkinson's disease that you can really appreciate how nice it is that we have properly functioning basal ganglia. That's really what today's lecture is about, although we won't talk about them in any great detail. The basal ganglia are your filter. They are the brain nuclei that make the decision of whether or not we should do something. And in Parkinson's disease, when we have the loss of dopamine neurons, the decision-making abilities of the basal ganglia is going to suffer. And what you'll see is the inability to appropriately and swiftly select actions. Here we can see a gentleman getting out of bed, and this is probably taking longer than it takes most of us. That's because he's suffering from Parkinson's disease. That slowed movement is typical for Parkinson's patients. What we're going to do is review what Parkinson's disease looks like, what causes it, and what we can do to treat it. The treatment options are a whole lot better than for Alzheimer's disease, but unfortunately none of them are disease modifying. When you think of Parkinson's disease, I want you to think of a poverty of movement. That's primarily what Parkinson's disease involves. So at its heart, it's a motor disorder characterized by slowed movements, that would be the bradykinesia. Also, in later stages, an absence of movement, the akinesia. Rigidity and resting tremor are often going to co-occur, and these will all work together to create gait disturbances. You should have a look at the two brain slices in this image. They're both showing us the midbrain. The brain on the left is from a control patient, and you'll notice two nice dark strips. They're full of some black substance, and that's why we call it the substantia nigra. That's all that means. So, I can recreate this. Here's your midbrain. You have your corticospinal tracts running in here, kind of nifty. Right in here, we see these darkly colored neurons. And they're darkly colored because they're making neuromelanin. It's melanin, it's a colored pigment that's made by neurons. And it happens to be made by dopamine neurons in the substantia nigra, pars compacta. So the SNC is something you need to tuck in your head. These are the neurons that degenerate in Parkinson's disease. These are dopamine neurons. Dopamine neurons in the SNC degenerate profoundly in Parkinson's disease. So compare the brain on the left to the brain on the right. The brain on the right is from a Parkinson's patient, and you'll notice that the black substance is now a faint gray substance. The loss of those dopamine neurons is what's going to cause the dysfunction in the basal ganglia and lead to the inability to properly and swiftly initiate action. As the disease progresses, the loss of dopamine neurons will then lead to cognitive and mood disturbances. But the first symptoms to arise are going to be motor. So PD is primarily a motor disease. Compare this to Alzheimer's, where it's primarily a disease of memory, and motor impairments only come on at the end stage of the disease. Now, AD and PD are the two most common neurodegenerative disorders. AD being the most common, PD coming in second place here. So it's fairly common, and as with Alzheimer's disease, it's a progressive disorder, meaning that it's going to get worse over time. The motor symptoms that arise early on will progressively worsen, and as the disease moves on, new symptoms will arise. You can read the incidence, and you can see in the graph below that the prevalence of PD increases with age. Not surprisingly, it's caused by the death of neurons, and we never make any new neurons. So over time, you can only have an increase in neuron death. It's never undone. The incidence of PD is about twice as high in men as in women, and the overwhelming majority of cases don't have a family history, just like in Alzheimer's disease. There's a little bit more heritability in PD. It's about 15% as opposed to only about 1% for AD. The core features of Parkinson's disease would be slowed, low amplitude movements, and difficulties initiating movements. And that's kind of how the disease starts and eventually gets to. So bradykinesia is going to come on earlier. That's the slowed movement. This will affect limbs. It will also affect face as well. So that facial bradykinesia creates what we call the masked face, where there's sort of an absence of emotion in the patient's face while they're executing actions. And that's because it's difficulty to move the facial muscles appropriately. There's also mood disturbances, subtle mood disturbances in the beginning of the disease that could also affect how expressive our face is. Now you'll notice here the hands are also slowed. His left is affected more profoundly than the right. You'll notice he can move his right hand a bit, and the left is kind of locked in place. That unilateral severity is not uncommon. It's not uncommon for the disease to start on one side and then spread to both, but still more profoundly affect one side. In addition to slowed movements, the amplitude of movements can be much smaller. So this would be the hypokinesia that's going to create that typical shuffling gait, or the Parkinson's shuffle, where you have very small steps, little shuffles. And you'll notice he kind of stops and right there, fails to initiate the next step. And that failure to initiate movements would be akinesia. And you can think of brady, hypo, and akinesia as being sort of different severities of the same thing. That is, the inability to appropriately carry out movements. It starts with slowness and lower amplitude and eventually progresses to a complete absence. Now, in addition to those core features, we should see at least one of the following. That would be rigidity. You can see here this gentleman is showing some rigidity to that passive movement of his wrist there. Same thing is true at the elbow. So we can see rigidity, resting tremor. Here we can see the unilateral onset. So the left hand there is exhibiting a little bit of a resting tremor. This is different from an intention tremor, where when you point or try to grab something, the tremor comes on. But in this case, he's just resting his hands on his legs, and his left has a 4 to 6 hertz tremor. Of course, PD patients are a fall risk because of their slow, low amplitude, or absent movements. It becomes difficult for them to prevent falls and to correct any imbalance. In later stages of the disease, you're going to see this postural instability because you need to have more profound slowing and absence of movement. Because of the treatments that we use for Parkinson's disease, you'll also see dystonia in some cases, where you have these involuntary movements taking place. That's because of the treatments. Because we're losing dopamine neurons, one of the treatments that we do is to bump up dopamine levels. Since the loss of dopamine leads to an absence of movement, an increase in dopamine leads to an increase in movement. And that would be dystonia. Now, if it's going to be Parkinson's disease as opposed to some other form of Parkinsonism, there has to be degeneration of dopamine neurons. If we don't see degeneration of dopamine neurons, we don't see responsiveness to dopamine-based therapies, it's not Parkinson's disease. There could be a lesion somewhere in the basal ganglia that creates those motor abnormalities, or it could just be some sort of drug-induced Parkinsonism. So in order to confirm the loss of dopamine neurons without having to take a brain slice, we can just take a gander using PET. There's a variety of PET ligands available to look at dopamine transporters, or the vesicular monoamine transporter that's used to put dopamine in the vesicles. So we have a variety of tools there to look at dopamine neurons without having to crack open anyone's skull. And here's a nice example. This is called DAT scan. This happens to be SPECT imaging, which is kind of like PET, just with a little poorer resolution. In either way, you put a radioactive ligand on that's going to stick to dopamine transporters. Dopamine transporters are going to be present on dopamine neurons. So the dopamine neurons here that are projecting up into the basal ganglia and spitting out dopamine, they have dopamine transporters on their axons. As dopamine neurons die, the amount of dopamine transporters decreases. So when we put on some sort of PET ligand or SPECT ligand that's going to bind to the dopamine transporter and give us some signal, we're going to see reduced binding and reduced signal. And that's exactly what this image is showing us. So on the left, healthy age-matched control, you can see two sort of comma-shaped structures in this DAT scan. So here's the brain. We're looking at the caudate there. They kind of look like commas. They're symmetric on both sides. When you look at the PD patient, they're calling it hemi because it's only affecting one side right now. You'll notice that one of those commas is losing its tail. That is indeed the tail of the caudate that's suffering from a loss of dopamine neurons. When the dopamine neurons die, no more dopamine transporters, no more ligand binding. So the motor symptoms that are characteristic of Parkinson's disease, along with imaging data to support the loss of dopamine neurons, is strongly suggestive of Parkinson's disease. Very high sensitivity and specificity, both of which around 98%. Pretty good tests. Neuroimaging can also be useful for ruling out other potential issues, like just some lesion somewhere in the basal ganglia that disrupts our ability to appropriately carry out actions. Along with the motor symptoms that we see in PD, there are non-motor symptoms. Dopamine neurons are going to project throughout our central nervous system. And a major target is going to be the basal ganglia, but we hit other places as well. And dopamine is going to contribute to our ability to properly regulate our autonomic nervous system, properly regulate our thoughts, emotions. And so you'll see a host of symptoms that aren't related to action, selection, and initiation. And these non-motor symptoms appear to have a much more profound effect on quality of life. And that's what I've put in this table here. So I've listed the non-motor symptoms. There's 4 there, and then there's motor impairments on the bottom, so we have some kind of comparison. How well does the severity of these non-motor symptoms correlate with quality of life compared to motor symptoms? And you can see the correlation coefficient is much lower for motor impairments. Undoubtedly, they suck, but not as bad as the autonomic dysregulation, the inability to properly control your bladder or bowels. That is definitely going to reduce your quality of life. Sleep disturbances will certainly reduce quality of life. And the mood disturbances that come on will undoubtedly decrease the quality of life. So just a quick summary on the diagnostic criteria here. The main things that we're looking for would be that bradykinesia, hypokinesia, akinesia, resting tremor, some rigidity. Either one of those is good enough. In later stages of the disease, postural instability is going to make Parkinson's patients a fall risk. And if it's going to be Parkinson's disease compared to any other Parkinsonism, we have to see the loss of dopamine neurons and responsiveness to dopamine therapy. Later in this lecture, we are going to see responsiveness to dopamine therapy, so you get an idea of what I'm talking about. These are going to interact with each other and some other symptoms to create other potential clinical features. Again, the onset may be unilateral, only affecting one side of the body first. That hypokinesia, whenever walking, will create the shuffling gait. PD patients tend to be stooped over, and with the dopamine replacement therapy, you can see dystonia. Now, the loss of dopamine neurons is going to create the symptoms of Parkinson's disease by affecting the proper function of the basal ganglia, our filter. So how do we determine whether or not we're going to do something? Now, of course, motor output is ultimately going to be carried along that corticospinal tract. Now, this cartoon is not anatomically correct, but that's okay. It's too good to be true, which is probably better than too true to be good. But you get a good idea of where the basal ganglia are, and that is down below the cortex. That's why they're called the basal ganglia. I don't know why they're called the ganglia, because they're in the central nervous system, and some folks do call them the basal nuclei, which is more correct, but we're going to be wrong because everyone else is. We're going to call them the basal ganglia. So in this illustration, you want to look at striatum, the GP, the STN. Those are components of the basal ganglia, and you notice they're right there deep in the center near the thalamus. So before we stimulate upper motor neurons to stimulate lower motor neurons and do something, whatever that is, our cortex first checks. It doesn't just act all on its own. We have checks and balances in our central nervous system, and ideally in our government as well, but maybe not so much today. The planned actions from our cortex are going to be passed down from the cortex to the thalamus. And really, the cortex and the thalamus are communicating with each other all day, every day. They carry out what we call the corticothalamic loop. And this is fundamental for our conscious awareness. Whenever you disrupt this communication, you lose consciousness, and you can rapidly snap animals out of anesthesia by stimulating certain thalamic nuclei to then waken the cortex. So the cortex and thalamus are always talking to each other. Now the thalamus does what really any wise entity would do. When they're asked, is this a good idea? They say, I don't know, ask someone else. And the someone else that the thalamus asks is the basal ganglia. So the cortex has a plan. It says, thalamus, is this a good idea? Thalamus says, I don't know, let's ask the basal ganglia. And the basal ganglia has really one of two answers, yes or no. The yes comes along the direct pathway. And we'll think of the direct pathway as being excitatory. So if it's a good idea, the basal ganglia act along the direct pathway to stimulate the thalamus. That's a yes. Now, we're not going to break apart the different components of the basal ganglia until next semester. So for right now, we're going to treat them as a black box, or on the slides, a dark blue box. Doesn't matter. BG, basal ganglia. Here's my cortex. Here's my thalamus. Here's my thalamo-cortical loop. This is what keeps me awake. My cortex says, is this a good idea? We'll get our answer from the thalamus in a bit. If the basal ganglia think this is a good idea, the direct pathway says, go. If it's not a good idea, or when we need to terminate an action, the indirect pathway inhibits the thalamus. So when the cortex sends excitatory input to the thalamus approves by sending excitatory input back, so that those upper motor neurons then fire action potentials and stimulate their appropriate lower motor neurons. So we need some excitatory input from the thalamus. By stimulating the thalamus, the direct pathway says, go. By inhibiting the thalamus, the indirect pathway says, no go. So far, we haven't talked about dopamine neurons. Let's correct that. Dopamine is going to regulate the balance of direct and indirect pathway output by acting on both of them. So, in the SNC, we have dopamine neurons. And these dopamine neurons are going to provide input into the basal ganglia. So let's put an indirect pathway neuron up on the board and a direct pathway neuron. Now both of these neurons have dopamine receptors. The direct pathway neurons have what we call D1 dopamine receptors. And what that means to you for right now is that it's going to be excitatory. That's going to depolarize my direct pathway. The indirect pathway neurons, on the other hand, have D2 dopamine receptors. And what that means to you for now is that that doesn't depolarize the neuron. It might subtly hyperpolarize it, but it definitely doesn't cause it to fire action potentials. Think of it as inhibitory. So dopamine, which is going to stimulate both of these receptors, is going to then excite direct pathway and inhibit the indirect pathway. So when we have dopaminergic input, we let off the brake and we mash the gas. This is going to excite the thalamus, essentially approving the message. If we approve the message, we send excitatory input to the cortex. So those upper motor neurons fire their action potentials, release glutamate to stimulate the lower motor neurons, and then those lower motor neurons go release their acetylcholine to cause muscle contractions. And we do stuff. We move around. But before we do that, before we activate our upper motor neurons, we have to have approval from the basal ganglia. In Parkinson's disease, when we have that profound degeneration of dopamine neurons, no more go. No more no-go. No more inhibition of no-go. So now, rather than getting an increase in excitation and a decrease in inhibition, because we've lost that dopamine, well, we're not stimulating go, so this is gone, and we're not inhibiting no-go, so this inhibition is gone. So the inhibition of the inhibition is gone, so we get an increase in inhibition of the thalamus. Now the message is, no, don't do that. Whatever it is you're thinking is a bad idea. So we get a decrease in excitatory feedback. We don't fire action potentials. We don't stimulate our lower motor neurons. They don't release acetylcholine onto muscles, and we don't do stuff. Here's a gentleman failing to do stuff. Although he has the bright idea of getting up out of his chair, his ability to approve that action has been impaired by the loss of dopamine neurons. So his planned actions get passed into the basal ganglia, and because of the lack of dopamine neurons, they're unable to be approved. So the best that he can come up with are these slow, low amplitude movements that are characteristic of Parkinson's disease. Now the cause of dopamine neuron loss is multifactorial, but they all kind of have the same thing in common, and that's metabolic stress. Dopamine neurons are always active. So these neurons are what we call pacemakers, meaning they're going to fire action potentials all day. They're always releasing dopamine to create some sort of basal tone of dopamine. This is a fine idea. This is great because if you have tonic dopamine, let's think about this. So the concentration of dopamine is on the Y, and this is time. It doesn't really matter. Because we're not at zero, we could have both an increase or a decrease in dopamine. But think if dopamine neurons didn't pacemake, if they were at 0, we could only have an increase. There'd be no way of ever lowering dopamine levels if dopamine neurons didn't pacemake. But dopamine neurons do. Because they pacemake, there's always some level of dopamine present at the synapse. So now they can signal either an increase or a decrease by increasing or decreasing their firing rate. So it makes them a little more flexible. They're able to send 2 different types of messages, not just one. And again, this is a fine idea. Where they've failed is with how they pacemake. Dopamine neurons are going to use voltage-gated calcium channels to drive their pacemaking. In fact, they use L-type voltage-gated calcium channels, and that L stands for long. These are calcium channels that are open for long periods of time. We've covered what happens whenever we have a chronic elevation of intracellular calcium. That leads to apoptosis. So dopamine neurons have to spend an enormous amount of ATP to clean up that calcium. And if they don't, they're going to experience apoptosis. We'll see what happens whenever they don't clean up that calcium pretty soon. Now what this illustration is showing us is another reason why dopamine neurons are fragile. Dopamine neurons live in the midbrain. They live in about 2 areas, the ventral tegmental area and the substantia nigra. So if we think about where our midbrain is relative to everything else, here's the midbrain, thalamus, and we got our big giant cortex here. So here's my dopamine neurons, and I need to get dopamine here. Each individual dopamine neuron is thus going to have an enormous terminal field, or volume that its axon occupies. So, that big terminal field, terminal of course being the axon, and the field being the area that it covers, means that each dopamine neuron has a whole lot of membrane. So when it fires an action potential, it moves a whole lot of ions. So every time it fires an action potential, it has to spend a whole lot of ATP. When it synthesizes that ATP, that can lead to free radicals, or what we call reactive oxygen species. Those free radicals can then damage proteins, lipids, DNA, etc., and create that oxidative stress. We think that metabolic stress is so problematic to dopamine neurons in part because of the heritable mutations that cause Parkinson's disease. These all, for the most part, they have in common that they affect mitochondrial function, and they don't improve it. So by decreasing mitochondrial function, you essentially start off with a little bit of oxidative stress, just when everything is working perfectly. Now let's take into account that large terminal field of dopamine neurons. If you look at this illustration, what it's showing you is the axon of one dopamine neuron. It branches, and they color one branch black, one branch red, but all the coloration in that illustration is from a single neuron. So it goes from the midbrain up into the striatum and covers it basically top to bottom, front to back. So this is an illustration of its large terminal field. Big terminal field, using calcium to pacemake. That's a recipe for disaster. Disaster mediated by metabolic stress. We also think that metabolic stress plays a role in Parkinson's disease because of the MPTP story. So this is in the notes. You can give it a read. But back in the 80s in Northern California, there was a heroin analog called MPTP that people were making. And these at-home chemists didn't always do a perfect job. So they were people, just like all of us, and people make mistakes. And the mistake that they made was rather than creating this synthetic heroin, they created MPTP, which is the precursor for MPP, which is an electron transport chain inhibitor. The electron transport chain is, of course, that chain of electron transporting proteins in the mitochondria that's absolutely critical for ATP synthesis. When you inhibit the electron transport chain, those electrons that are moving along it have nowhere to go but out of the electron transport chain to increase the production of free radicals. This is devastating to dopamine neurons because MPTP is taken up selectively by dopamine neurons. So when folks thought they were taking heroin, they were actually taking MPTP. That MPTP was taken into their dopamine neurons, metabolized to form MPP. It then inhibited mitochondrial function, stalled the electron transport chain. The high levels of free radicals that were produced then led to profound dopamine neuron loss, and overnight they became Parkinson's patients. You can see the effects of MPTP in the illustration on the bottom. Compare the left to the right. The left, we can see far more black substance. The substantia nigra has a lot of dopamine neurons in it compared to the right. Mice treated with MPTP suffer the same consequence as humans. And this is one of the models that we use for Parkinson's disease. We just inject MPTP into the SNC and degenerate one half of the brain. So we essentially have a built-in control in these mice. One half normal, one half Parkinson's. Another feature that you'll see in Parkinson's patients is the presence of Lewy bodies. These are essentially the amyloid of Parkinson's disease. They're not made of A-beta, they're made of alpha-synuclein, so it's a different protein. It has a different distribution than A-beta, and we also don't really know what it does. The alpha-synuclein probably acts in the presynaptic terminal, so maybe what happens is depletion of the presynaptic sites. Alternatively, alpha-synuclein might just make a bunch of holes in the membrane. We don't know. But we do know that dopamine neurons can't break down alpha-synuclein. Because dopamine neurons can't break down alpha-synuclein, alpha-synuclein then builds up in dopamine neurons. So we get these Lewy bodies forming within dopamine neurons. However it kills them, whether it be through forming pores in the membrane, that of course is going to cause a loss of membrane potential. So their membrane potential will approach 0. That's going to lead to excitotoxicity, just like it has in pretty much every lecture in this class. Or maybe alpha-synuclein just depletes all the vesicles in the presynaptic sites. Then dopamine neurons don't work anymore. So whether it kills them or just makes them not work doesn't really matter. The end product is the same. Inappropriate dopamine input to the basal ganglia and the inability to properly select actions. So what do we do about it? Well, in the initial stages, what we're going to do is try to replace that dopamine, and in later stages, what we're going to do is try to correct the network abnormalities. First, just a bit on how we score Parkinson's disease severity. This is a questionnaire you may very well use someday. So we have the Unified Parkinson's Disease Rating Scale. This is going to be composed of a questionnaire as well as a motor exam. And in the motor exam, they're going to be looking for tremor, rigidity, bradykinesia, posture, standing. For rigidity, we're just looking for nice, smooth, passive movements. In this case, in the head. They'll check the limbs as well. And they're going to do this on and off medication to see how the disease is progressing. Because when on medication, this can mask motor impairments. So here we can see the bradykinesia test. You can go ahead and do it at home, tons of fun. So just tap your finger as quickly as you can, and if you'll go back several slides, you'll see Parkinson's patients do this much slower. On to the treatments. So the first line of therapy is dopamine replacement. So in the initial stages of the disease, the first thing we do is either give dopamine receptor agonists, or we give the precursor to dopamine, or we do both. Now the precursor to dopamine is called L-DOPA, or levodopa. This is taken orally with meals a few times a day. And L-DOPA is going to be converted to dopamine in a one-step reaction by an enzyme called dopa carboxylase. Along with L-DOPA, we also administer carbidopa. Carbidopa is an inhibitor of dopa carboxylase. The reason that we give this when taken orally is because L-DOPA can cross the blood-brain barrier. Dopamine can't. So if we convert L-DOPA to dopamine before it has made it into the central nervous system, it's not going to make it there, and it would be a waste. So patients will take L-DOPA and carbidopa. The carbidopa is going to inhibit that peripheral dopa carboxylase so that we're not making dopamine in our blood, but instead we're transporting that L-DOPA throughout our blood. It crosses into the central nervous system, and then dopamine neurons take it up and create dopamine. Parkinson's patients have to respond to dopamine therapy by definition, and here's a gentleman who's going to respond. Now, whenever we do motor exams, and by we I mean not me, but maybe you, we do this on and off meds. So here we're seeing him in the off phase. This gentleman's task is to walk across his yard, turn around, and come back. Now you'll notice they've edited for time. That's because this is not a swift task like it should be. Notice right there, a little Parkinsonian shuffle as he's trying to turn around, those low amplitude, kind of slow shuffling movements. And he's going to make his way back. 30 minutes later, after taking his L-DOPA, now he's on the on phase, and it's subtle, but I think you'll notice the difference. You'll notice rapidly walking across the yard, large full steps, a little bit of dyskinesia in the head there, but far better motor function. This is temporary though, so you'll notice he was off. He's been taking carbidopa. He's going to metabolize it. We're not affecting the underlying issue, which is the loss of dopamine neurons. So he's going to have to continue to take his dopamine replacement therapy, and his responsiveness to it is invariably going to decrease with time. As dopamine neurons continue to degenerate, he's going to need higher and higher doses. We're not modifying the course of the disease. We're providing some temporary relief, but this temporary relief is much better than we saw in Alzheimer's disease. In later stages of the disease, we're going to have to resort to deep brain stimulation, which is where electrodes are implanted into the brain, and then they pass electrical currents to the nearby neurons to stimulate them. Now, based on the frequency of stimulation, this can actually lead to a decrease in neuron activity. By hyperstimulating them, you don't allow them to properly repolarize so they can't fire another action potential. So even though it's called deep brain stimulation, in some cases it's actually deep brain inhibition, and that's likely what it is in this case. So while the patient's awake, electrodes are going to be implanted into their basal ganglia. And the goal here is to electrically correct the abnormalities that have occurred because of the loss of dopamine neurons. What we want to do is allow that direct pathway to do its job again. Now, deep brain stimulation is not disease modifying either, but it is very helpful in later stages of the disease, at least in terms of controlling motor abnormalities. So here, this gentleman has the deep brain stimulator implanted, so he has the electrodes in place. They're going to turn the stimulator on now and then dial it so that it appropriately controls the abnormal motor function. And you can see his resting tremor is now controlled. They're going to do a quick test to make sure he doesn't have bradykinesia. Ask him how he feels. And now you can see he can quickly execute his actions there. Deep brain stimulation doesn't modify the course of the disease, just like dopamine replacement. It does improve responsiveness to dopamine therapy, though. So with the deep brain stimulator, you won't need as high of doses of dopamine replacement. The issue with deep brain stimulation, and this is subject to debate, is that it can exacerbate the non-motor symptoms, such as the cognitive impairments that occur and mood disturbances. And there are some reports that it's associated with an increase in suicidal ideation and suicide attempts. And then there are other reports that say that that's not really true, and that it only happens if the electrodes are improperly placed. So this is certainly subject to debate. Undoubtedly, deep brain stimulation can help with the motor symptoms, and there's an unclear story right now about whether or not it can make the non-motor symptoms worse, which, remember, those are going to have a more profound effect on quality of life. So before we get together, review the review questions, review the notes, and if you have any questions along the way, send me an email or fill out the questions box on the class website. I'll see you later.